Okay, we're next going to go to my good friend Jack Tuszynski at the University of Alberta. There he is. Good morning, Jack. Good to see you. And he's going to talk on the biophysical properties of microtubules and their functional significance for neurons. Jack, take it away, please.
Thank you very much, Stuart, and hello, everybody, and thanks for inviting me to speak to this audience. It's very interesting. I want to say at the beginning that the word revolution calls for something very ambitious, and I'm kind of a modest guy. I would actually go for evolution. And so I'll start with some aspects of neurophysiology which are important to me as a physicist, namely the importance of electricity, electrical currents driving our brain and its cognitive functions. And as a physicist, I'm really interested in this, but I would like to go deeper. And this is the evolutionary aspect of my talk, namely beyond the Hodgkin-Huxley model of action potentials and into the cytoskeleton, of course, microtubules, which are in the title, and other structures. Hodgkin-Huxley model of the action potential involves membranes of neurons and ions, but neglects the cytoskeleton and ions. And I'd like to, in this talk, convince you that there is a very intricate interplay and incredibly sensitive dependence on ionic concentrations, on pH, also on the frequency of oscillations, electromagnetic oscillations. So this is the sort of main theme. I will also probably recapitulate some of the aspects presented by previous speakers, namely Arat and Travis and Stuart and others who talked about microtubules. The outline of my talk is given here. I will start with a very brief overview of the structure and function of tubulin microtubules, and then I'll move to the topic of electrostatic properties and the conductivity, ionic conductivity along microtubules, including capacitance, and finally responses to electromagnetic fields, which may lead us to the sort of main mystery and the main intrigue in this story, namely quantum excitations, possibility and significance. The list of my collaborators is very long, but these are the most important ones. Arat and Greg, Travis, Aristide, and of course Stuart, with whom we've been collaborating for close to 3 decades, I believe, and my colleagues at the University of Alberta. So let's start with the structure and function. You've seen and heard quite a bit, so I'll be brief here. Microtubules are important in all eukaryotic cells for creating the morphology of the cell, for motility, for transport inside the cells, but probably even more. And the aspects of or the role of microtubules in neurons is one of the most interesting and not quite properly explored properties. Here we have kind of a standard neuronal signal propagation, down propagation, that is from the receptors, sensory receptors through axons and synapses to other neurons, and with the dendritic activation of something happening in the interacting neuron. But the treatment of the neuron as a binary off and on switch is kind of an affront and definitely not sophisticated enough to account for the neurons and other cells' intelligence, and the cytoskeleton is implicated in all the, not all the, but many functions of these cells. And in particular, it is well known that rearrangement of cytoskeletal matrix is taking place during learning through various cascades of interactions involving calcium waves and the breakdown of the microtubules and reassembly of microtubules, as well as transport of various molecular components. In the interest of time, I will not be able to go into great details, but this is what Travis proposed in one of his papers, which is the opposite direction, neuronal signal up propagation from actually the molecular level in interactions involving reactive oxygen species produced by mitochondria, affecting tubulin, which then produces excitons, which propagate along microtubules, and then magic happens. I guess we don't know exactly how that gives rise to global microtubule-like state change that may then be sensed by the neuronal membrane, and somehow neurons talking to each other would understand the reorganization taking place inside the cytoskeleton. There are a lot of dots to be connected here, but I think the basis is quite well articulated, and I think quite a lot of what was said in this regard in this conference about the quantum aspects of that kind of story. I'll come back to it at the end, but before we do that, I really think that there's a lot of detail that needs to be fleshed out in terms of how all of this happens in the particular milieu. And this is a reconstruction of the microtubule itself, with the cylindrical structure and the C-termini sticking out, these peptide chains, which play, in my opinion, a very important role, almost like antennae, because they have a lot of electrostatic charges on them, and they can sense even subtle electromagnetic field differences as well as mechanical waves. So you heard already about how tubulin is the building block of microtubules, the alpha beta heterodimer that is kind of a brick from which microtubules as hollow cylinders are constructed by each eukaryotic cell. But what is really important to state at the outset is that this is not just structure proteins, with mechanical properties which are non-trivial, quite rigid, and at the same time dynamic, which is almost an oxymoron, but also very intriguing electrical and electromagnetic properties, which require further clarification in terms of the biological function. I will mention here the changeable resistance and capacitance, the action of a voltage pulse transmission line, memristance, which means these are memory containing devices, if you can call it this way, as well as very sensitive receptors of electromagnetic influences. And we haven't really explored even this surface of that story, but I will say a few words about each of these topics, in particular how microtubules respond to electromagnetic waves in different ranges, different frequencies and energy levels. So this is incredibly multi-layered and multifaceted, and I think it merits a lot of research. And one of the reasons for this, of course, as we heard yesterday, is, of course, involvement of microtubules in pretty much all neurodegenerative diseases, including a special Alzheimer's disease, where we heard about the role of tauopathy, MAP-tau, that interacts with microtubules and forms a scaffold, which kind of disassembles and forms neurofibrillary tangles. Now, we've done some simulations ourselves recently due to the ability of constructing atomic resolution models of microtubules with MAP-tau. As you can see here, this is from recent crystallographic data, and it's quite intricate. MAP-tau forming a tight connection on the outer surface of microtubules, and this is actual electrostatic map of the same MAP-tau structure from the protein data bank with the ID shown here. The red patches are negatively charged parts of MAP-tau, the blue ones are positively charged, and white are neutral. So there's a huge amount of electrostatic interaction, and as we heard, hyperphosphorylation of MAP-tau leads to this pathological transformation eventually, with a downstream effect of neuronal death. And And this is our unpublished preliminary data. Some of you who are interested in Alzheimer's could contact me later on and we could discuss it or maybe collaborate, where different serine residues on the map tau are ranked here and according to their electrostatic energy. So it's not the same significance for each serine because some of them have very little electrostatic free energy. So targeting particular parts of map tau for stabilization, for example, or preventing hyperphosphorylation would be important. So electrostatics is, in my opinion, important to understand in this connection. So that leads us as a segue to the next section of my talk, electrostatic properties of tubulin microtubules. And suffice it to say that tubulin is an absolute outlier in the family of thousands of proteins that the human body produces. It has a very high net negative electric charge, typically, 40% of which is focused on the C termini. These histograms show you with the arrow where tubulin is, sort of outside of the main distribution of proteins according to charge on the left and dipole moment on the right. They carry a very high charge. They carry also a very large permanent dipole moment due to the amino acid construction and composition. And therefore, they will be incredibly sensitive to everything interacting with this, namely electric fields, voltage changes, as well as electromagnetic fields. So this is another view of a microtubule on the left. Now, this is a very old but very important paper in the literature on this topic, where you can see the charge distribution on a microtubule and the two ends, panels B and C. That gives then an asymmetric aspect of electrostatic properties of microtubules. One end is more negative than the other. And the surface is also largely negative, but with patches of positive charge, which, without going into too much detail, offers a richness of behavior of this C termini. They can interact there, as well as other molecules with dipole moments or charges. Okay, so this is, maybe I'll just go quickly through this. This is about polarizability. Polarizability is response of a physical object to electric fields, and that's change in the dipole moment as a ratio of the electric field change. And there are several aspects of polarizability in tubulin, and that includes the inherent dipole moment, as well as movable dipole moments related to the counter ions surrounding charges on the residues, which are charged, and this finally C termini movement, and the water molecule is around. So it's a complex problem, but it's definitely measurable. And we, in collaboration with Professor Aristide Dogario from the University of Central Florida, we did measurements, and that shows you the first kind of intriguing aspect of it, both the polarizability on the left and the dipole moment on the right in black, and the net charge on tubulin are pH dependent. So the environment of this inside the cell, if it changes pH, as is known, for example, to take place in neurodegenerative diseases, it will affect the response of microtubules and tubulin to electric fields. So let's move on to the next topic, which is now moving charges, namely ionic conduction, which is what I started from in terms of Hodgkin-Hatchley equation. That's ionic charge movement across membranes, but of course they didn't go deeper and did not explore how the cytoskeleton responds to the ions around it, and how these ions can be transported from place to place inside the cell. And many types of ions, of course, exist. Typically, sodium and potassium is the highest concentration, sodium second, and then, of course, protons, all of which are positively charged, and they are attracted to the surface of microtubules. And we've done measurements, which I'll very briefly go over, that indicate in amazingly non-trivial conductivity of ions along microtubules. Microtubules can both improve and hinder ionic conductivity depending on the concentration of ions, depending on pH, depending on protein concentration, etc., they can also form simple obstacles. So the story started in 2006, where we published a paper with Horacio Cantiello. At that time, he was at Harvard, and we reported some very unusual effects. Microtubules actually amplifying ionic pulses in terms of the voltage created by them, and acted as molecular transistors, which was very, very unusual and still is. We then also explored actin filaments, which I will not talk about, but actin filaments had the properties of diode-like behavior. But microtubules amplify these signals, and that's worth mentioning. We created models, electrical cable models of microtubules with these predicted values of the capacitance, resistance, and inductance. And that was 2008, and it took almost 15 years to do further experiments. And Arat Kalra in my lab and later at Princeton refined these measurements and provided some body of factual information about how microtubules behave in terms of conductivity as biological wires and capacitors at the same time under different conditions. Here we have a recent paper from the group of Horacio Cantiello, who is now back in Buenos Aires, and showing you that the microtubules also support self-sustained electrical oscillations in the roughly speaking 40 hertz range, low frequency, but corresponding to the brain waves. So that's very intriguing, whether the external waves are acting on microtubules inside neurons and vice versa, if there's some sort of deeper level of signaling taking place in terms of electromagnetic oscillations. So now on to the measurements that we did in Edmonton with Dr. Shankar's collaboration. These are micrographs of microtubules and the different concentrations. Some of that looks like spaghetti, but they form networks. These networks are actually electrically active, and I think that also offers some future prospects for hybrid structures, maybe some devices that could be implantable, and creating some computer-like. I know that Stuart doesn't like to talk about the brain as a computer. I don't either, but we can actually do neuromorphic computing using these structures as templates. So these are the results of some of the measurements we did. We first of all found that the conductivity of microtubules is greater at low ionic concentrations than that of tubulin itself, and also is frequency dependent, peaking at about 100 kilohertz, which is a separate story, at least the potential treatment of cancer, brain cancer in particular, which has been recently clinically demonstrated. And then we measured capacitance. So it's a storage device, charge storage device, at the same time as being a conducting device. And microtubules at low ionic concentrations have a greater capacitance than free tubulin, and resistance at low frequencies changes to high frequencies. At low frequencies, microtubules are more resistive than tubulin, than solution, vice versa at high frequencies. So everything is kind of subtly complicated, but not completely impossible to model or understand mathematically. So at low ionic concentrations and higher frequency values, microtubules decrease solutions impedance or increase conductivity of the, if you were to talk about cytoplasm, then they would be the preferred pathways for conductivity or conducting signals. At high ionic concentrations, the opposite happens, and microtubules become more resistive. So that's kind of what the cells may, I don't know if they do, but they may utilize in terms of switching the role from charge storage devices to charge transport devices, and creating some sort of logical connections. And these are the tabulated results of the extrapolated values for the resistance capacitance and inductance of microtubules, individual microtubules for a 20 micrometer long microtubule as an example. And all of this is published, so I don't ask you to remember these numbers, but you can look it up. And the experimental demonstration of this, which was published in about within the last two, 3 years, kind of corresponds neatly. It's not too far off from the theoretical prediction that was done in 2006 or 7, so almost 15 years ago or so. Now, we also did computer simulations of all possible concentration values of ions, as well as distance dependence from the surface of microtubules and the C-termini, and we found that the electrostatic potential really changes depending on the ionic concentration, and there is a crossover at some point. So the question was, at what point do microtubules switch from, let's say, being better conductors than the cytoplasm and become less conducting, more capacitive, depending on the ionic conditions. And so we found something around 100 millimolar concentration of potassium, which is right around what typical cell physiology tells us. So the cells could also, with small changes of ionic concentrations, turn the microtubule function from conductive to capacitive, or vice versa. So that's kind of interesting for the future. And now let's move on to the next topic, not quite last, but I think we're getting there, response to electromagnetic fields. So I talked so far about electric fields, electrostatics, electroconductive, ionic conductivity. Now let's move on to electromagnetic. So things like infrared or terahertz radiation, visible, ultraviolet. You probably heard a lot about ultraviolet. You'll hear again in a minute or two. But there are some frequency-specific responses which are kind of summarized here in terms of both mechanical, electromagnetic waves, depending on the structure. Perhaps Anubhav mentioned some of that. So different mechanisms respond to different frequencies. And so just to summarize in one sentence, microtubules can in fact be multi-channel devices. What I mean by this is that they can propagate signals at different frequencies through different channels, either through mechanical or ionic or electronic or conformational or excitonic. And that kind of opens up a Pandora's box of possibilities. Since about 2007 or so, it was known that microtubules respond to electric fields in terms of their alignment, drift, and even polymerization. These were some gliding assays showing that as you apply electric fields, they start aligning. So this is 50 volts per centimeter electric fields, and you can see how microtubules align themselves nicely. But the story, the plot thickens, as they say, because all of this depends also on the frequency, not just intensity of the electric fields. They realign, they reorient, they drift. Here you have a panel showing you how different frequencies lead to different geometrical orientation of microtubules, which are shown in white in this diagram with the two electrodes generating the fields, and different mechanisms in the environment have been measured by electrophoresis, electroosmotic flows, and thermal conduction, all conspiring to this somewhat complex behavior. So I guess, again, the emphasis is on complexity and being very careful as to the conditions, ionic, pH, temperature, as well as electric field intensities and frequencies. So we have a huge space of for possibilities of different responses. So as a physicist, I really want to know, whether we're comparing apples and apples or apples and oranges. These are our own little videos Kara made, how microtubules and tubulin respond to the changing polarity of the electric field. And what was also discovered is the role of pH. So at low pH, microtubules actually become positively charged, which is a big change from what I said in the beginning, highly negative to begin with. But if you lower pH, then they absorb, they get protonated. Histidines get protonated, and the net charge on the microtubule becomes positive. So maybe this is not so important physiologically, but I think technologically in the future it could be very important, as you could manipulate these computing devices made out of microtubules into switching from negative to positive, and vice versa. Now, the next set of experiments was done with Professor Hegman and Cam Howe at the University of Alberta, and we demonstrated here that microtubules can be completely demolished or disassembled, disintegrated by exposure to terahertz electromagnetic fields. Terahertz fields are kind of ubiquitous. Water absorbs a lot of energy, and you can use them in, and they have been used extensively in airport scanners. If you go through security, you're exposed to it, but perhaps not for long enough to disassemble your brain microtubules, I hope. This is a short video kind of summarizing 15 minutes of exposure to terahertz fields by microtubules in solution. They were shown in red, red on the black background, and they kind of melt away on exposure to terahertz fields over 15 minutes. This is compressed into a few seconds. Okay, so there's that, and the most sensitive frequency to which microtubules are is about 0.5 terahertz, so low terahertz. I don't know what frequencies are used for airport scanners, but hopefully not these. We also did computer modeling of what happens to microtubules exposed to electric fields, and they are dynamically rearranging their dipoles, realigning the permanent dipole, and also C-termini start fluctuating, and that probably explains the instability of microtubules when exposed to electromagnetic fields. Let me just finish this electromagnetic story with the experiments, the results of experiments done in Florida, again by the team of Professor Dogariu, who showed that this is now in the visible range. So light, laser light, was used to show that microtubules can be, when they polymerize or build from tubulin, they can be slowed down, and actually less of them are made when exposed to light. So light, in this case, promotes depolymerization, is destabilizing or slower microtubule growth, if you will. Okay, so now let's spend the last few minutes on the probably the most interesting topic, and has been probably discussed a little bit by both Travis Craddock and Kara and Stewart: quantum excitations, other quantum excitations, microtubules, and what type of excitations, what do they do, and this kind of thing. Before we actually go to these recent experiments, I want to bring back this topic of whether microtubules can be memory storage devices. And we published a paper now more than a decade ago, which computationally predicted that you can encode a huge amount of information in microtubule serine residues on C-termini by calmodulin kinase phosphorylation. And this is from that paper, which I'm very proud of. This is still speculative, but I think has a lot of merit. So in a way, this could be called quantum information, if you could, because this is quantized, and each bit of information possibly can be become a qubit if the state is in a superposition, quantum superposition form. All right, so excitation transfer in tubulin in the quantum realm was predicted in our paper from 2014 that tubulin has 8 tryptophans in the dimer, which resemble the photosynthetic systems, FMO molecules, which create a quantum state. So we assume that this might be happening in microtubules, and it took about a decade again to show that this indeed is happening in experimentally. And these were simulations from Travis, more recent, on the propagation of these quantum excitations traveling over several dimers along the microtubules. They could be probably re-energized by another quantum of energy and propagate farther. And these are the experiments that Arad performed in Professor Scholz's lab at Princeton. Tryptophan fluorescence, using very clever assays showing both the lifetime of excitations and their spatial extent, using the AMCA molecules shown in the middle that absorb and emitted fluorescence from tryptophans, and we could distinguish the two. And by doing so at different AMCA concentrations, Arad was able to predict how far the quantum states extend, beyond which there is no communication, so to speak, between interacting tryptophans. And the results are shown here. In addition to this question, the important issue of anesthetic quenching of these excitations was raised and answered. And the short summary of these results is that the lifetime of these quantum excitations is somewhere between 2 and 5 nanoseconds, and the radial or spatial extent of this quantum state is about 6 nanometers or so. So the size of a dimer, roughly speaking, not greater than that, probably, unless we can't really say that it's not connecting 2 dimers. But so these are substantial time scales and spatial extents, although probably not sufficient for a physiological relevance in the context of neurophysiology. We still have a bit of a gap. We would like to go to a millisecond, but time will, I emphasized in the first part of the talk the importance of the environment. And I think that may change these answers substantially depending on the environmental conditions in terms of ionic concentrations, pH, and maybe connectivity between microtubules, inclusion of maps, etc., etc. And anesthetics actually reduce the lifetimes by about 25% or 20-25%. So the predictions made by Stewart and Sir Roger Penrose are some, I would say, supportive in general, but specifically still leave a little bit of room for improvement, to put it this way. However, the second group of experiments, I'll be done in a minute, so I'm very aware of the time, was performed at the Dogario lab at the UCF Orlando in Florida, with the excitation using a laser in the 650 nanometer wavelength. The parameters are given here. The intensity of light was not too high. It was kind of comparable to biological luminescence effects. And the time, the lifetimes were measured for tubulins in buffer solution, microtubules in buffer solution. This is unpublished yet, but it'll be soon published. And the results were quite interesting. We compared tubulin to microtubules to the buffer, and microtubules exhibited the longest lifetimes compared to tubulin as well as buffer. And also the anesthetic molecules lowered this lifetime. Now, the good news is, the really, really good news is that in this case, the lifetime, both of tubulin and microtubules, was in the hundreds of milliseconds. In fact, for microtubules, it reached almost half a second. So that's incredibly encouraging. And also the fact that light in this range. The wavelength 650 nanometers has a much lower energy than ultraviolet range that was used for the Princeton experiment, so it's more realistic from the biological point of view. The somewhat open issue is still of the amount of photons which were emitted right after exposure over, let's say, picosecond to nanosecond scale in subradiant, superradiant part. The subradiant, which was long-lived, those 400 millisecond or so events, this was maybe less than 1% or around 1% of the photons were long-lived. So it's a technical issue. It's not a be-all and end-all unsolvable problem. But I just want to highlight it as something that needs to be looked at to be able to bring it to more realistic scenarios. So in summary, I want to say that the importance of microtubules in the context of neurophysiology cannot be overstated, and also the importance of technical details in terms of environmental conditions, as well as the diversity of behaviors, capacitive, resistive, conductive, etc., is super important, and the response to electromagnetic fields. Okay, and Stuart, last slide. Allow me to summarize the implications for OR. The experimental findings are supportive, but still require further refinement. We need maybe somewhat longer lifetimes and longer ranges. Visible light promises better outcomes. I actually would like to go into infrared, if I may, because there I think we could probably go into even better numerical results. So thank you very much for your attention. I'm sorry I've talked for more than allowed time.
All right, thank you very much, Jack. Let's have one question from Dimitris Panatsis, and then we're running into the break.
Great talk. Thank you so much. Two quick questions. Would you expect that electric activity of microtubules could be picked up? How big could it be? And could it be picked up with, electrodes that we use in neuroscience? I don't know, local field potentials or EEG?
Yes. I believe so. Yes, and in fact, this is the first experiment I mentioned with Horacio Cantillon in 2006, where there were two micropipettes that send the signals, and they definitely picked up the activity of an individual microtubule. I mean, in electrophysiology of neurons, you have thousands of microtubules in bundles. So I really totally expect that this is a doable experiment. And I mean, if somebody could do this, I'd be super happy.
Yep.
I'm not a neuron sort of experimentalist, but I think people who work with neurons, patch clamp, crowd and so on, could easily do this, in my opinion.
And following up on this, would you have from your theory, do we get any predictions about how changes in electric or electrostatic or properties of microtubules, right, due to polymerization or other processes, changes in solvent pH, how would this affect function of neurons or neural populations? So, for example, you alluded to memory, right, and memristor. Yes. And we know that at a large scale there are oscillations, for example, oscillatory behavior. Perhaps action potentials could slow or change and see gamma oscillations. Do we have any predictions that connect scales?
Well, this is the open issue. Yes, I don't think there are many predictions connecting the scales, but I think for me this is the most pressing topic, making connections between the scales from tubulin to microtubules. We've done that from microtubules to the neuron. It has not been done, and except maybe for some individual papers in the past. I wrote a paper in the 1990s with my student Andrew Brown on how neuronal action potentials could affect microtubules. So a long time ago. There is a potential interaction both ways.
Thank you so much.
For sure. Yep. You're welcome.
Okay, we got to wrap it up, Jack. Thanks so much. Great seeing you. Good work. And I'll be in touch. We'll have a break and come back
at 11.